Hello everyone, this is Nobod Sharma to present our research title Efficient Segmentation Techniques for the Automated Detection of Acute Lymphoblastic Leukemia in Microscopic Images. We all know that blood is one of the most essential components in our body. It contains 4 components. White blood cell is one of them. White blood cell plays an important role in the immune system of our body, and leukemia is one of the deadliest diseases in human life that affects WBCs, which may indicate the sign of blood cancer. For detecting leukemia, image segmentation is helping a lot. Now, what is image segmentation? Image segmentation is computer vision task that involves dividing an image into multiple segments based on certain characteristics such as color, intensity, texture, boundaries, etc. We applied some image segmentation method for detecting leukocytes and comparing the performance in image dataset. In present time, leukemia can be diagnosed by CBC, which is complete blood count, bone marrow aspiration, pathological test, and aspiration biopsy. The laboratory test for leukemia is quite monotonous and takes more time. In the manual examination of this test, trained pathologists count the blood cell one by one from the microscopic blood smear images. So this process can lead the variability in diagnosis and error in counting blood cell. That's why it needs to be automated, and our research work will solve this problem. We reviewed lots of papers which are related with image processing, and from this we find some image pre-processing and segmentation method which are mostly used in our reviewed paper. For our research work, we use image grayscaling, median filter, and morphological operation for image pre-processing, and watershed method, Wood's binarization method, and KNAS detector are used for image segmentation. Here in the right side, you can see the pie chart where they display the usage ratio of the segmentation method of our reviewed paper. Now one paper is from Lawrence Puzzo. Their research focuses identification and classification of blood cell images. They use histogram equalization and threshold based segmentation, and the outcome and limitation of their paper is that the identification of WBC is quite well in some cases, but the worst result is overlapping leukocyte cells, but they give more attention in classification rather than the identification. In the right side, you can see the performance evaluation table that is from their paper. And the second paper is from Nurul Hazwani, AVD Halim. They focus on automatic counting the blood cells using image processing technique. For this, they use HSB color space segmentation method, and their system is only detect accurately and work well on less amount of blood cells, but couldn't count the overlapped and large amount of cells. In the right side, the performance evaluation table from their work also. Our goal is to speed up and decrease the error in diagnosis system for acute lymphoblastic leukemia. For this, we use some refined image pre-processing technique and some segmentation method to build the appropriate automatic system to count the white blood cell and detect leukemia. The outcome of our research work that we find out the best segmentation method for the automated detection of leukemia, and the impact of our research work that stand as a step toward automating and enhancing the performance of ALL detection in comparison with present diagnosis systems. For our research work, we use most commonly used dataset ALL-IDB, which has 2 subsets, ALL-IDB1 and ALL-IDB2. Both subsets has leukemia and non-leukemia cell images. ALL-IDB1 has 108 images and ALL-IDB2 has 260 images in total. But the problem is ALL-IDB has no ground truth data. So the question is, what is ground truth? The ground truth is a term of machine learning. It refers the correct or true answer of a specific problem that can be used to compare and evaluate the model result. For evaluating our models, we manually create the ground truth of each image using ImageJ tool. The methodology, which is divided into 5 parts: image acquisition, image pre-processing, image segmentation, automatic WBC count, and performance evaluation. For image acquisition, we use ALL-IDB dataset, and then after we have done image pre-processing, we split it into 3 parts. First, we convert every RGB image into a grayscale, then image filtering. For this, we applied median filter, and lastly, using morphological operation for removing the noises. After that, we took the processed image into the image segmentation part. We applied 3 segmentation method: watershed method, Wood's method, and KNAS detector. After segmentation, we perform a counting operation to evaluate the result. For performance evaluation, for this 3 segmentation method based on accuracy, error, precision, recall, and F1 score. This is the example of grayscale image. This is the median filtered image, and this is the morphological image. These 3 images are the image pre-processing part, and next image is the example of segmentation part. This is the watershed method, and this is the Wood's method, and this is the KNAS detector. Now the experimental results part. We can see there is an assessment table of the segmentation methods. In this research work, we use 50 image samples from ALL-IDB1 and ALL-IDB2 because we manually made the ground truth of every single images, and it is more complex and time consuming. In this assessment table shows the 3 segmentation methods counted cell of every images. These are the experimental results of each segmentation method. From watershed methods, we got 91.09% accuracy and 8.91% error, 99.72% precision, and 82.34% recall. From Wood's method, we got 90.95% accuracy and 9.05% error, 98.90% precision, and 82.34% recall. From the KNH detector, we got 85.11% accuracy and 14.89% error, 94.92% precision, and 77.06% recall. In F1 score, the watershed method achieved 90.20%, Utsus binarization and KNH detector achieved 89.86% and 85.06% respectively. Along with that, we also noticed a table for total performance analysis of this three segmentation method, where you can see the watershed method perform much better compared to other methods. Among them, the KNH detector performed the odds in our experiment. First of all, we are going to enrich the dataset to explore more models and try to enhance the detection accuracy. Also, we plan to deploy a system where the blood cells are automatically masked. We get this motivation during our research work because we faced some issues when we didn't find the ground truth of our dataset. For this, we have done this manually. That's why we plan to work on this project, which may help upcoming researchers who are going to work on this field. Along with this, we try to develop a web-based automatic WBC counting system, which provide a major advantage, especially to pathologists. The conclusion of our research work is that we found watershed method to get higher accuracy, which is 91.09%, and in segmenting the overlapped cell, watershed method worked done very well compared to other segmentation methods. The finding is most suitable for the segmenting the leukemia cell and developing a computational model for automated detection of ALL. Now these are the reference of our research work. Thanks to Intex Research Lab for providing us the technical support and giving the opportunity to utilize their resources, and thank you everyone.